Hello everyone, you're most welcome to the Universe Science Scholars Academy, also known as USSA. So, today's lesson I would like us to look at something that is a little bit interesting as far as the study of science is concerned. But before we dive into it, I would like us to a little bit give a highlight on what USSA or the Universe Science Scholars Academy is all about. Now, at the Universe Science Scholars Academy, we are basically a platform that is located in Uganda, offering online classes specifically via Zoom, Google Meet, and we do offer them in the different science subjects that are in relation to physics, biology, chemistry, but we also do offer the logical bit of it, which is the study of math, and with key interest also look at ICT. That is a little bit so much important for a learner as they grow up into a better scientist in the universe. So we also do give free consultation lessons, also mentorship classes, and give academic discussion sessions specifically with our learners, and that we do offer in our WhatsApp channel and also the distributed WhatsApp groups that the students are located. You can also find us in our other platforms. For example, you can find us over X, also get us on TikTok, also get us on Instagram, also you can locate us on Snapchat. Alright, and also find us on Facebook. Specifically, our social media handles will have the name that is USSA. And today I come with a little bit of a question that I'm asking all the science students that are around the universe. Have you ever wondered, or rather asked yourself one of the things, and one of the kind of things that I'm talking about right now, specifically, why human beings produce eggs? Okay, why do human beings produce eggs or sperms? Specifically in humans, the sperms are produced, we all know that the sperms are basically produced by the male people, and the eggs, you can also call them the ova, are also seen being produced in the females. Alright, have you ever wondered how that comes about? Or evenly why children resemble their parents, but still in the resemblance, they are unique in their own different attributes. And when I talk about the attributes, those are specifically the characteristics that make up that individual, or rather the children. It could be the skin color. It could also be something to deal with the height of the person. There is all that different outlook, but then they still look alike. Okay, I know most of you have ever wondered, wow, how is that so? I expect myself, yeah, since we resemble, we have to be reasoning alike and everything. But why isn't it so? Or rather, how chromosome numbers stay constant generation after generation. Let's take a very key example. In humans specifically, we see that we are having a little bit of chromosomes, which are 46 chromosomes in a cell. All right, but why are these chromosomes always the same from generation to generation? We are not having them either doubling to give us a little bit of 92 chromosomes. No, we always have them consistent from generation to generation. Why is that so? So in today's video, we are going to be answering all these questions in particular. But what does that create in our brains? That actually gives us a little bit of a new concept that let's dive into understanding these different concepts specifically. That is relating to the study of meiosis. How amazing it is to be studying meiosis at a moment. So most of the people have been looking at this different prospect that is relating to the study of meiosis. You've been asking yourself, wow, all the questions that you've been wondering about how do the children still remain to look unique despite the fact that they're actually being born by the parents? why do they see that there is a constant chromosome number? All right, and why do you specifically keep on seeing to it that we're having the eggs or the sperms being produced? How does that happen? Meiosis is going to be answering that all. So I'm so much delighted to be taking us through meiosis quite at a moment. Like I said, my name is Andrew Desire. Most of the people like to call me Master Andrew. So specifically, we are going to be diving in and looking at what meiosis is all about today. All right. And without further ado, let's be looking at what brought us here specifically. And let's look at this. But before we go there so much, I would like us to understand something. This video is actually created to make you understand what meiosis is, the stages that are involved in meiosis, why meiosis is important, and how it actually creates genetic variation. So do not leave this video without understanding all these prospects, because they are expounded and deeply explained in this video. That's what we are going to be looking at a moment right now. So please stick around and be there till the very end. So let's go further. So before we start looking at meiosis as a whole, there are some specific keywords that we are going to be meeting, or rather interacting with, as far as the study of meiosis is concerned. All right. But then I cannot really dive in and move on and give us different explanations of the different work without you people knowing specifically what each of the different terms specifically that we are going to be meeting in the near future stand for. I hope that is actually something that you are going to be taking into due consideration at a moment. So let's dive in specifically, looking at one at a time. So you normally ask yourself that what is a chromosome specifically? So a chromosome, we can look at it just as a thread-like structure that is actually carrying the genetic material it itself. And where is it located? This is actually located and seen around the nucleus. Around the nucleus. Alright? And that will dive in further to look at what a chromatid is. So normally when we look at a chromosome itself is a structure. We'll be illustrating it so much as just specifically something of a sort. Alright, with a center in the middle here. So I would like us to name the different parts of this that we are given as a chromosome. So specifically, when we are having one strand of this, like this actually forms what we call a chromatid. So this itself is a chromatid. Then the two of them will be forming what we call the sister chromatids that are located on one chromosome. While the center here of attachment of the two chromatids is normally what we refer to as the centromere. As the centromere, as simple as it is. So when we move down here, we also look at a keyword that is homologous chromosomes. What are homologous chromosomes? These are specifically chromosomes carrying the same genes, all right? Chromosomes that are carrying the same genes. That's what we call the homologous chromosomes. And moving down a little bit to look at some other simple terms that we normally see. And without further ado, we are looking at something that is called the chiasma. So normally when we have homologous chromosomes that come together and pair up, for example, we can have this as a chromosome of itself. Then we also have another one that we could be illustrating here as that, right? So when the two actually come up and they get so close, normally there is what we call crossing over that occurs. Okay, and what is crossing over? Crossing over itself is just exchange of a specific portion of the genetic material between two non-sister homologous chromatids. So we can have it like this. We are having this at a time. All right. Then we can also have the other chromosome that we did illustrate earlier on, and we're having it like this. All right. So at this specific point that we see that these specific chromosomes are crossing over, okay, portions of the chromatids are actually crossing over. This specific point where a crossing over is occurring is what we refer to as our chiasma. In plural, we normally call them the chiasmata. Then when we look down here, we're looking at a term that is called a bivalent. A bivalent itself is just a pair, okay? It's just paired up homologous chromosomes. Like here, when we have these two that are paired up like this is actually called a bivalent. And most of the people like to refer to them as tetraends, which is also okay. Then synapsis. Synapsis itself is just literally the process by which the two chromosomes come together and join. When we're having independent a chromosome here and another one here, the process as to which they come together and join is what we are trying to refer to as synapsis, a process by which homologous chromosomes pair up, as simple as it is. So with that small insight of the keywords that we have been having, let's dive into the specific prospect that has brought us here, specifically right now about in the moment, and that is looking at something to deal with. What is meiosis? Meiosis itself is just a type of cell division that produces haploid reproductive cells. Alright? Haploid reproductive cells. And here specifically we are looking at the eggs, as we said previously, and at the same time we're also looking at the sperms. Alright? Normally also called the gametes. That's why we call it aka the gametes. All of them, the same name can be used. Alright? So when we're looking at meiosis, meiosis is just a type of cell division. Or rather we can say the splitting of a cell. And we do know that there are basically two types of meiosis. To talk about the very first one that we have been looking at, and that is meiosis itself is one of a kind. Alright? Then the other form of cell division is what we call the mitosis. So mitosis itself in humans occurs so much in the somatic cells. Alright? And somatic cells here, that is just the body cells, and that is not in the reproductive cells themselves. And I hope that has built a little bit of an understanding to you about what meiosis is all about. So let's be moving on and see a little bit of other prospects that we are going to be looking at right now. And that is something to deal with the importance of meiosis. Why is meiosis a little bit so important at this specific moment? So when we look at meiosis itself, the very first point that we are going to be looking at here is that meiosis itself is responsible, or rather leads to production of gametes that are needed, or rather involved in sexual reproduction. And in humans we normally have said that we are calling those the sperms in the male, right? And also not forgetting that we are having the ova or the eggs in the female. Alright, so these are the two specific forms of cells that we're going to be looking at, and specifically how the sperms themselves are formed. The sperms are formed in a process that we normally refer to as spermatogenesis. How amazing it is to pronounce the terms. And these eggs are normally also formed in a process that we refer to as oogenesis. Alright? Oogenesis, as beautiful as it is, and pronunciation is so much key at this specific level. So let's be moving at the different points also. We also realize that if we look at the second point, it says that we are having the point. It also is responsible for maintenance of constant chromosome number in species. Like we said initially, that when we're having the in humans we normally say that we are having 46 chromosomes. So if we had these 46 chromosomes, remember that upon fertilization specifically, we are having chromosomes that are being brought up by the maternal side and the ones that are brought up from the paternal side uniting together from the egg and and the ova. So if we had in mind that we are having the egg having 46 chromosomes, and then we are also having the sperm itself having 46 chromosomes. So if these two unite in a process that we refer to as fertilization, you realize that we will be forming a cell that is having a little bit more number of chromosomes that are adding up to about 92. But this doesn't occur. So we are so amazing. It's so amazing to realize that we are having the 46 chromosomes number that is in a cell always maintained from generation to generation, specifically by the fact that we are having meiosis occurring, because meiosis is the one that's responsible for dividing this chromosome number into the 23 and also the 23 that are seen in the ova and the egg. So when the two reunite, we have our 46 chromosomes formed back again in our zygote. See how beautiful it is? So let's look at the third point here that we have. And the third point here is specifically dealing with the fact that meiosis itself is the one that is also responsible, or rather creates what we call genetic variation. Let's highlight this. Genetic variation, making every individual a little bit kind of unique. When variation occurs, when we say that we are having variation has occurred, this is definitely just the difference in characteristics per individual. That is what you can understand by the term variation. So when we say that variation has occurred, that means the characteristics per individuals has a little bit been unique. You're having different characteristics. You have different characteristics from that of any other person that you may be knowing around you. And all of that is because of meiosis. And we are going to be diving further to look at how specifically meiosis creates what we call genetic variation. And finally, we also have a point here that says that it is also meiosis that is responsible for supporting evolution. Without meiosis, we would not be having evolution going much far as it is right now in different species. So that is a little bit of something that you need to be knowing as far as the importance of meiosis is concerned. Then we will be moving up and looking at specifically the specific divisions that are involved in meiosis. And we know very well that meiosis itself is divided into two major divisions, and that is meiosis 1 and meiosis 2. Alright? And understanding the different prospects that we are having in meiosis 1 and meiosis 2 is so much key. And meiosis 2 is so much key and important for you as a learner in the Universe Science Scholars Academy. So meiosis 1 itself is targeted to separation of the homologous chromosomes. Okay, this one separates the homologous chromosomes. And at this specific moment, I expect everyone to be knowing what the word homologous chromosomes means, because you already explained them. Then the meiosis 2 is targeting to what we call the separation of the sister chromatids. This one specifically separates the sister chromatids. And all of them are so much important as far as the study of meiosis is concerned. So let us be diving into our very first prospect that we are going to be looking at, and that is something to deal with the stages that are involved in meiosis itself. So normally what you need to be having in your head that before meiosis starts, what normally happens is that we have a little bit of a preparation stage that occurs, and we normally call it interphase. This can also be called the preparation stage, like we have a little bit named it before that. So what normally happens at the stage of interphase is that in this stage of interphase, we normally have the DNA itself replicating. All right, DNA replication occurs at this specific angle. So let's look at this. We are having a cell, all right, and I told you earlier on that we are having In the cell, we have the nucleus, and let's dive in specifically into the nucleus where we're having the chromosomes located. So we do know that we're having 40, we have about 46 chromosomes that are seen per cell in normally, that is in the human beings. So we are looking at something that we're having. We are just going to be taking, for example, just two chromosomes that are inside. We are just going to be taking two that are inside. All right. So normally, what happens in this specific prospect that we are having before having this all happening, we are just having in the nucleus just chromosomes, paternal and maternal chromosomes can be seen inside here. All right, can be seen inside here. But what we say that we are going to be having the DNA itself replicating, and at the same time also the chromosomes duplicate. So this specific chromosome at a time will duplicate to form what we call the chromatids. Now that we are having two sister chromatids, as you can see here specifically, like I've tried to illustrate here as it is there. All right, and also there is increase in energy here, and at the same time we're having organelles that are seen in the cell replicating. All right, or rather that the organelles themselves will keep on multiplying at a specific moment. So here we are having each chromosome forming what we call the sister chromatids majorly, and that is actually joined by the centromere that we are seeing here. That is the centromere. All right, so that is what you need to be knowing as far as interface is concerned. Then the key thing that normally happens, or rather the one that I need everyone here to put key much attention into, is something to that is actually the other stages that are seen common in both divisions. Both divisions meaning that we are seeing it happening in meiosis 1 and also happening in meiosis 2. Right? And the very first one we're going to be looking at is prophase, followed by metaphase, followed by anaphase, then finally telophase. So I do like my students to always remember that by just what we call the math. All right? So this is happening twice in all the stages of meiosis. I hope that has been understood. So let's dive in right now and look at specifically meiosis 1. So in meiosis 1, the very first stage like we talked about is what we call the prophase 1 that is going to be occurring. And this is normally attributed or rather known to be the longest, all right? The longest and most important stage that actually occurs during it is the longest and the most important stage that occurs during meiosis. All right, so meiosis 1 itself is normally characterized by a target that happens that is actually also called as reduced depending reduction division. Some people also call it reduction division. All right, which is all correct. And this stage normally, like we said previously, that it is actually a stage that reduces the chromosome number from what we call the diploid to the haploid, and we are going to be looking at how that actually occurs. That's why it's also called the reduction division itself. So moving into prophase, we said that the very first thing that you need to be knowing that happens during prophase 1 is that normally we see the chromosomes condensing and the shorten becoming so much visible. All right, that is the first thing that occurs during prophase 1. All right, then we also have the homologous chromosomes pairing up, and that is a process that we called synapsis. Later on, we actually explained what it is. And when synapsis occurs, we all know that we are having the bivalents or tetrans being formed. At this specific extent, we are having the bivalents, sometimes also called the tetrans, being formed here specifically when we are having synapsis has already occurred. And it is also characterized, the prophase 1 is also characterized by the cell, or rather the nuclear membrane disappearing. Here specifically the nuclear membrane is also seen to disappear. Okay, the nuclear membrane disappears specifically. And at the end of it all, we normally have what we call the formation of the spindle fibers. So when we talk about the spindle fibers, this is something that I need everyone to be jotting or rather understanding deeper. Here we are going to be looking at specifically the animal and the specifically going to be looking at the animal and the plant cells that we are going to be looking at right now. But then before we go there further, I need you people to understand something, that when we say spindle fibers form, in animals specifically, the spindle fibers are formed by what we call the centrioles. But there is something that we need to be knowing, that in the plant cells we don't have the centrioles. So where do we have the spindle fibers being formed from? Or rather, the spindle fibers are formed by what we call the microtubule organizing centers. Specifically, that's where we are having our spindle fibers being formed from. And characteristic to this very process, the most final and important thing that happens in process one is what we call crossing over. And crossing over, like we say, is just when we are having the non-sister chromatids exchanging the genetic material at a specific portion that it actually takes up. Like we say that this one is actually characterized at, it starts occurring what we call the chiasma. Alright, that we illustrated last time, that we are having a chromosome like that and another chromosome also here like this. So we said that at this specific point here, we're already having what we call crossing over occurring, and it occurs around here. So I hope we have understood. Not forgetting that this is actually inside a cell. Alright then, so let's move on to study specifically the different substages that are seen occurring in what we call process 1. So process 1 is just something according to what we have explained. Alright, according to what we have explained, you should be in position to understand that there are actually substages that occur in process 1, explaining the different things that we have seen occurring above. And normally I do like to tell my learners to have acronyms on how to remember some few things. And normally I like to abbreviate this as LZPD squared, representing leptotent, zygotent, pachytent, diplotent, and what we call the diakinesis. So looking at one at a time, when we look at what we call the leptotent, the leptotent itself is just where we're having the chromosomes become thin and visible. Okay, they become visible. That what happens specifically during the leptotent stage. Then the zygotent stage is where we're having the homologous chromosomes pairing up, and the pachytent itself we are having crossing over occurring here. This is where we're having crossing over occurring. It occurs during the pachytent stage. Then the diplotent is where we are having the homologous chromosomes beginning to separate. Alright, separate. We are going to be indicating how all of this happens specifically using vivid examples and drawings that we are going to be having in this specific video. Then diakinesis is characterized specifically when the spindle fibers are fully formed. When we have the spindle fibers fully formed, that is actually what we say that we are having diakinesis has occurred. I hope you've understood the few sub stages that are involved in process 1. So allow me to let us, or rather usher us into our next page that is involved in what we call the metaphase 1. So here in metaphase 1, what normally happens is that we have the homologous chromosomes pairing up at the spindle equator, and that specific arrangement of these chromosomes at the spindle equator, we are having the cell, and we are having chromosomes specifically coming to align themselves. We are having a division that is occurring here at the center. We could just have it like that. So when we are having this coming around and aligning around the equator of the spindle, this is what we are calling specifically this form of arrangement. When this homologous chromosomes come and line around is what we are calling the independent assortment. This is actually a random orientation of these chromosomes, or the homologous pair of chromosomes, at the equator, or rather the equator of the spindle, which is this. This is the equator of the spindle. While these other locations around here and here are what we call the poles. The poles. So that is what happens during our metaphase 1, that we are having a little bit of our bivalence arranging at the equator. This is a bivalent arranging at the equator. The spindle fibers actually get attached to the centromeres. Now this is it. We are having here at this specific point, we are taking now the animal cell, we are having the centrioles there. So these centrioles literally produce what we call the spindle fibers. So the spindle fibers come and attach at the centromere. All of us know that the centromere basically is actually a point that joins the two sister chromatids together. So when we have the spindle fibers attaching at the centromeres, that is actually something or other characteristic of what we call metaphase 1. So that is all you need to be knowing as far as metaphase 1 is concerned, and we are going to be diving into the next that we are calling anaphase 1. So anaphase 1 it itself, characteristic to it is that we normally see the homologous chromosomes starting to separate. Initially, we remember that we ended metaphase 1 with the spindle fibers attaching themselves to the centromeres. But what happens here in anaphase, that we have the chromosomes separating, okay, and they'll be moving to opposite poles specifically. Alright, they'll be moving into the opposite poles. So this actually is characterized by the separation of the chromosomes, and this is actually pulling them into two haploid sets of chromosomes, and each set that is actually seen located at the spindle fiber. Okay, not the spindle fiber specifically, but at the equator. So this is what we are having in right now. So this separation itself, it is actually what we are seeing, that this is actually why the chromosome number reduces, because we are having here, this is it. So we are having this initially, and we are having our centrals here. So in the next step, we are having this that has occurred, that we are having initially, we're having this, then we're having also this attaching themselves there. So but at this specific point, we are having specifically the chromosomes that have been actually pulled now to the different poles by the spindle fiber, right? That is actually produced by the centrioles. So that is what we are trying to mean by the processes that occur during anaphase 1. So at this specific extent, we say that this actually reduces the chromosome number by a half, because we are having this being pulled to the other pole, and this other side also we are having the chromosomes being pulled to the other pole, and that will let us enter into the next stage that we are calling telophase 1. So telophase 1 itself is where we are having specifically the chromosomes have reached the different poles, and the nuclear membrane it itself will also reform. Here we have the reformation of the nuclear membrane it itself. And if we are to have that a little bit into drawing, we can have it like this, right? We can have it like this. So at this specific point of telophase 1, it is actually, we are having what we call cytokinesis occurring. When we say cytokinesis itself is just something to mean the division of the cytoplasm. That is what cytokinesis is. So to indicate the division of the cytoplasm, we can just have something of a sort. Alright, then here we are having something that shows that we are having our nuclear membrane formed, and here also the nuclear membrane has reformed. And in here we're having chromosomes inside, alright? But then we are saying that they are now half because we are having each being pulled at the different poles. So we are going to be having this separating out. And the first stage it itself will be giving us what we call two daughter cells. Two daughter cells at the first stage. Alright? And after this specific point of interphase 1, there is normally a resting stage that occurs between meiosis 1 and meiosis 2, and that is what we call interkinesis. So interkinesis itself is a short resting stage between meiosis 1 and meiosis 2, and in this specific stage, there is no DNA replication occurring at this stage specifically. Sometimes we normally also call this interphase 2. There is no problem. Some people like to call it interphase 2. Interphase 2, right? But it is so prudent that you call it interkinesis. So with this specific knowledge that you have, allow us dive into our second division that occurs. So in meiosis 2, it is so important to know why does meiosis 2 occur. We could just be having meiosis as one, like just have the meiosis like we have the mitosis, okay, not having it being divided into meiosis 1 and 2. So it's very important that meiosis 2 occurs, in that remember that we are having in meiosis 1, we are having specifically the chromosomes themselves have been divided, the homologous chromosomes have been divided. But remember that we are having chromatids here, the two sister chromatids, but each chromatid itself is varying from the other in the sense that we are having already crossing over that occurred during our prophase 1. So it is actually necessary for this to occur. Why? Because it helps separate the sister chromatids, okay? It separates the sister chromatids. The sister chromatids it itself. Alright. Then it is also so much important that it occurs because at the end of the day, each gamete only gets to receive one chromatid from each chromosome. So if meiosis 2 does not occur, that will not happen. So it is so important in that each chromatid, each gamete receives only one chromatid from each chromosome. That is point number 2. Then we also have the fact that it actually helps the correct haploid chromosome number, okay, to be attained. If we are having in humans 23 as the haploid number, it is actually this meiosis 2 that causes that to happen. If it doesn't occur, then we would not be having that occurring. I hope that you have understood something. Like we also see here that specifically this meiosis 2 is so much similar to mitosis. Alright, so let's dive into the different stages that are involved in meiosis 2. And the very first stage that is involved in meiosis 2 is what we call the prophase 2. So prophase 2 it itself, here again we normally have the chromosomes becoming visible again, okay? The chromosomes become visible again. We also have the nucleoli specifically and the nuclear envelope dispersing. That means that we are having the structures of the nucleoli and the nuclear envelope disappearing from the cell, alright? From the cell. So we are going to be having these chromosomes again specifically open up a wide, and that is what happens. The also spindle fibers also are seen to appear, because remember that at the end of telophase 1 we had the spindle fibers disappearing. All right, the spindle fibers disappearing. I hope that I didn't tell you that in the previous explanation. So that is actually what happens there upon that specific form. So we are having the spindle fibers reforming specifically, then moving into the next. These are now just simple things as seen in mitosis. The next one is metaphase 2. So metaphase 2 is where we see the chromosomes again aligning at the equator of the spindle, not of but rather of the spindle, right? Of the spindle specifically. So what happens here during our metaphase 2 is that we are having here, now it is not the bivalents arranging themselves, but it is specifically one chromosome coming here. Let's say that we are having our spindle, the equator, moving like this. So we'll have just single chromosome coming to align themselves there. All right, coming to align themselves at the equator again. But then we are having the fact that we do remember that we already have the centromere there, and we are also having the spindle fibers attaching. Okay, like we said here, the spindle fibers attach to the centromere. All right, to the centromere itself. So at this specific stage, we will be moving to a different point that we are going to be calling the, what stage is next? All of us do know it is a stage that we call anaphase 2. Right, anaphase 2. So anaphase 2 here, normally we have the centromere splitting. I like to draw this. This centromere itself will split. And when the centromere splits, that means we are going to be having the independent chromatids now opened up. So when the independent chromatids are opened up, what happens is that we are going to be having the sister chromatids separating. We're already having the centromeres that have been already split, and then the spindle fibers will pull this independent chromatids towards the opposite poles. So we'll just have a single chromatid being pulled here, and also here we'll be having a chromatid just being pulled. The same to this pole. We are not pulling chromosomes this time around, but we are now pulling the chromatids themselves at this specific point of anaphase occurring. So that will specifically drive us to looking at the next state that occurs, and that is the state that we are calling the telophase. That is a little bit so common. So telophase 2 here specifically, the cells divide once more. All right, the cell divides once more, and also we would be having the nuclear membrane reforming here itself. We also have the nuclear membrane forming itself here again. All right, forming itself here again. So what happens is that by the end of meiosis 1, we formed the two specifically haploid cells. But then we are going to be having this at the end of, don't forget that at the end of telophase, we again having cytokinesis occurring, the cytoplasm splitting up. Okay, cytokinesis occurring. So here again, we are going to end up forming two. Each of these will form two, specifically two haploid cells. So by the end of at the end of this telophase 2, we are going to have 4 haploid daughter cells formed. Normally, we do like to indicate this as diploid and N as the haploid number. So this is N, 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 and all of these are actually 4 in number. So we know now that characteristic to meiosis is the result of formation of 4 haploid daughter cells. All right. So finally, you should be knowing that the final result of meiosis is that one diploid parent cell will be the one that is actually going to be producing, okay, 4 haploid cells. Okay, so this is actually what you need to be knowing as far as what meiosis is all about. Not just knowing it by definition, but have the whole picture of the whole of meiosis. When we say meiosis, what does it fall into? What does it talk about? And that specifically will be diving us into something that is a little bit nice, and that is something to deal with the study of genetic variation. Genetic variation itself is just specifically the difference in characteristics of individuals in a population. It could be in a species, depending. So the very first source of genetic variation here, we're looking at specifically the sources of genetic variation is crossing over. So crossing over here is where we had that exchange of gametes between the chromatids that was happening at a specific portion that we call the chiasma. So once we have the crossing over, that specific portion that has been swapped, because initially we had something of a sort, all right, something of a sort. So our final result, we are now forming a chromosome that actually looks like this. All right, chromosomes that actually look like this. How do they look specifically now? They now look like this. A portion of this is having a little bit of a segment of the other, and we also have a portion of the other having a little bit of a segment of the other along the chromatid. So you see that this is actually having genetic information carried from a different chromosome, and this one is also having genetic information in the chromatid carried from another chromosome. So that it itself would be saying that these specific chromosomes would have changed, and they are now varied because they are carrying genetic information that are different from the different chromosomes that they actually interchange during crossing over. I hope the point has gone home. Then secondly, the genetic variation itself also is arisen or rather caused by another source of genetic variation is independent assortment. That we try to explain that when we're having the chromosomes, okay, the chromosomes coming to align at the equator of the spindle. That doesn't happen dictatingly. Let's say that we are having specifically this, and that is the spindle. So we can have this chromosome maybe aligning with another chromosome here like this, right? Then also we can necessarily not say that we can have another chromosome here that can come and align with a different chromosome here. So they align independently. It's not necessarily that this kind of chromosome aligns itself with the similar chromosome there. So that means that at the point of their being pulled at the poles, you find that actually also happens at a random kind of way. So that is not dictated by the different attributes that are happening within the cell. So that actually makes you to understand that at the end of it all, we are having independent assortment also creating what we call genetic variation, because now we don't really, the characteristics that are bared by this chromosome is different by the ones that are paired here, and also different by this and this. So once we're having that being taken up by different cells, that means that we are having totally different genetic information carried in different cells at the end of meiosis. So that is carrying actually different characteristics that are responsible for the different characteristics rather that are going to be exposed by an individual at the end of it all upon fertilization. Then the fact that also there is random fertilization occurring, that is also where we are having a source of genetic variation occurring. Random fertilization, in that any egg can be fertilized by any sperm. Okay, any sperm can fertilize any egg. You don't have to dictate. When we're having the sperms moving along, normally in the male, I mean in the female, specifically moving along the fallopian tube, the very first one that actually is in position to meet the egg that was released from the ovary upon ovulation, that one will be the one that is lucky and will be leading to the formation of the zygote. So specifically, that is also a result of genetic variation, because now we are having specifically each sperm carrying different genetic information. Remember that these sperms were formed by meiosis, specifically under when we're looking at spermatogenesis. So when we have a sperm with different content, each of them having different content, that means that the zygote that was going to be formed when another sperm was actually going to unite with the ova upon fertilization is very different from the one that actually united with it formed. So that is another source of genetic variation. And let's be diving into to look at something a little bit important, that yes, when we have genetic variation occurring, there are also errors that are associated with it. Okay, the little bit of errors that are associated in meiosis, and that is what we are going to be looking at right now about in the moment. So normally when we are having non-disjunction occurring, non-disjunction is when we are having homologous chromosomes like this failing to separate. So at the end of it all, they are actually going to be pulled towards the pole as a whole because they have failed to separate. So this actually creates what we call the genetic disorders. Sometimes we normally call this as mutations specifically. All right, as mutations. And the very first form of mutation that we normally see that occurs is what we call the Down syndrome. All right. So Down syndrome itself is also referred as the trisomy 21, normally caused by an extra copy of a chromosome 21. All right. So in here, an individual has a chromosome number of about 47 specifically. All right. And having the different karyotype. Normally in female, we can be denoting it as maybe the 47XX, or we can have it as the 21 plus. All right. And the male being the 47XY or the plus 22. So we will be looking at that. Then also we are having another area that can occur. Specifically, that is the Turner syndrome. The Turner syndrome is seen or rather caused by a missing X and a missing sex chromosome. Okay? A missing sex chromosome. So here, instead of an individual having about 46 chromosomes, the individual ends up having 45 chromosomes, and the ones that are so much affected is the females. And this is actually seeing that instead of a female having XX, the female will be having XO because we're having a missing chromosome already there. Then the final one that we normally see, the other genetic disorder that can occur is what we call the Klinefelter syndrome. That is actually caused by an extra X chromosome in the male. Okay, so we are having a male having, instead of having XY, a person is seen having XXY. So this actually is attributed, normally these people are the ones that we normally call the transgenders or the feminists. Someone has more of the female characteristics that are actually being displayed. All right, so when you look at the Down syndrome, that is just an abnormality of chromosomes. Normally, these are just chromosomal mutations. Down syndrome is relating with an extra chromosome 21. Then we are having in the Turner syndrome, we have the missing X chromosome, and the Klinefelters, we normally have it as the extra X chromosome, specifically that occurs in the male. So there's something that I need everyone to be knowing, that yeah, there is actually no cure for these different disorders that are actually occurring in errors that are involved in meiosis, due to the fact that there is non-disjunction, failure of the homologous chromosomes to separate, right? So, but there actually are many treatments and support measures that are given to actually handle the specific symptoms, right, that are seen with these different individuals. Like the Down syndrome, someone who has the Down syndrome, normally they do give them the therapies, speech therapy, just having physical therapy. Also, they give them they normally go for regular checkups, all right? Then the Turner syndrome, normally the type of treatment that is given is we're having estrogen therapy, whereby we are having the person being injected by the estrogen. Because someone having the Turner syndrome, normally we have a person behaving more like a man. All right, like we have here, we're having someone having only one X chromosome in the female, so the person tends to have a little bit of less developed secondary sex characteristics. So we normally inject with the estrogen and help develop the female secondary characteristics, and also monitoring the treatment of the heart itself. All right, monitoring the treatment of the heart itself. So then someone who has the Klinefelter syndrome, normally we do have the testosterone replacement occurring. Okay, testosterone replacement therapies can be recommended. Also speech and educational behavioral support can be offered to these specific individuals. So that is a little bit of something that you need to be doing as far as the errors in meiosis are concerned. Yes, we do know that we are having meiosis occurring, right? But yes, there are also mistakes that happen, and we normally have to be grateful to God that you were born how you are. If it wasn't for him, who can maybe know you would be an individual with the Turner syndrome, or the Klinefelters, or the Downs. Who can tell? So always praise God for whatever that has actually happened to your life and how you look. It is so amazing and beautiful. People struggle with different problems that they can't reverse. Like if you had someone having Down syndrome, like we said, there is no possible cure. We can't pick the chromosomes and separate them. So you just remain that individual and you be grateful to God that you are one. All right? Since is our creator. So finally, our last is specifically our conclusion. Yeah. Did you understand something from whatever we have been talking about? Because initially we had our target that was rotating specifically for a learner to know what meiosis is. Do you know what meiosis is from the explanation that we have given? All right. Have you understood what it is and the different stages that are involved in meiosis specifically? And also, have you been in position to understand how genetic variation occurs? Have you been in position to know how important meiosis is? Alright? So those were the targets that we had for this video. And after understanding those different concepts, I hope that you did understand. What did you understand and how you're going to implement? So we are so happy. Okay, USSA is so happy that you've understood that different concept. But please don't forget to like our videos, share them, and comment. Right? This I remain Master Andrew or Andrew Desire at the Universe Science Scholars Academy, USSA. Let's explore the universe together. We love you. Thank you for watching.